Hello, everyone, and welcome to QIASeqsolutions for studying SARS-CoV-2 and its effects using next-generation sequencing. My name is Brian Dugan. I'm an Associate Director of Global Product Management for Genomics at QIAGEN. Everything we'll talk about today are intended for molecular biology applications. These products are not intended for the diagnosis, prevention, or treatment of a disease. So as we start thinking about tackling COVID-19, the SARS-CoV-2 virus, there's a whole host of different areas to look at. Primarily what everyone hears about often on the news is the need for diagnostics or better diagnostics. And this is really for rapid testing to determine the current status or previous SARS-CoV-2 infection. Beyond that, what we see is the need to look at it from an epidemiological point of view. And to do that, we have to have full characterization of the virus and also its environment from banked patient samples. And specifically, looking at the entire genome of the virus is interesting to be able to understand what's happening in clusters, what's also happening in different places around the world. The other thing that's also highly interesting is the need to look at an unbiased approach, like RNA sequencing, to understand not only where the virus is going, but how it passes through the body and where else does it go after it passes out of the body. But in addition to that, is also how the host or the human body actually responds to the pathogen. And so looking at host-pathogen interaction is extremely informational. Now, as we move beyond diagnostics to research applications, we know that there's four key areas that are essential to looking at the virus. The first, and probably most public, is for sequencing the virus for drug and vaccine development. Here we're looking at biomarker discovery of looking at the host, but also looking at the virus and making sure there's something that could not only treat patients in acute phases, but also be given prophylactically to patients to prevent them or to hopefully protect them from any type of infection. Now, in the public health space, sequencing the virus provides us the opportunity to look at viral evolution and also conduct contact tracing. As we move beyond that, what we're seeing is that there's interest in doing population screening. Now, most of the population screening that's talked about so far has been antibody-based tests to understand who has been exposed and who has not. But really, there will be a need in the future, potentially, to look at next-gen sequencing-based applications that can sequence thousands of samples at a time to understand if something has been infected from a surface to a patient to something else that you touch regularly to understand whether that virus is there. And finally, as we talk about the idea of the flu season coming up, there's also an interest in understanding what type of co-infections are also there as part of the SARS-CoV-2 viral infection. And so with that, there'll be a need to actually sequence the genomes or targets of multiple viruses in addition to SARS-CoV-2. Now, QIAGEN is tackling this on a few different fronts. And both a targeted and a non-targeted approach, QIAGEN has solutions that are available to really to be able to drive your research. And so if we start off talking about an unbiased viral RNA and metatranscriptome approach, QIAGEN has solutions essentially to be able to go from whether you're dealing with picograms or nanograms of input to over nanograms of input, depending on the sample type, to look at the global RNA expression of not only whether the virus is there, of how the host is actually responding to everything, and look at this as a totality. You can use this with our QIAseq stranded RNA kit, our FastSelect technology that removes a tremendous amount of ribosomal RNA, so you're really honing in on what's important in the sample. Our second approach that we have is virus-specific to SARS-CoV-2. And here it's an amplicon-based approach that is providing ultra-low input amounts in terms of what you're providing in terms of the front end, and now you're able to sequence the entire whole genome. This allows you to be able to do contact tracing, epidemiological studies, or other research applications that are focused on looking at how the virus is changing as it moves around the world. And finally, all this is tied together with QIAGEN's digital insight solutions. And whether you're looking at the basic analysis through CLC Genomics Workbench of looking for a report or variants, or even to do a phylogenetic analysis just for the virus, that's one option that you can certainly take. However, if you're looking at host response, you have the opportunity to actually look at QIAGEN's ingenuity pathway analysis to understand how the RNA expression is actually changing the pathways in the individual host. Our first part of what we're going to talk about here is looking at a targeted solution for tracking viral genome changes to support this epidemiological research. And to do this, we'll talk about just sequencing the SARS-CoV-2 genome. Essentially, it's a two-step process. One is a PCR amplification, and the other is followed by a library prep. So the first step is a cDNA synthesis, which is a random priming first-strand synthesis, and that then is followed by an enrichment. The viral RNA sample is taken, the cDNA synthesis is undergone, enrichment is done through a two-pull set, which is then pulled back together, and then gone through the rest of the reaction after quantification and normalization. Here you're able to take this entire reaction and now sequence the entire viral genome for SARS-CoV-2. It's essentially a 6-hour workflow. Now, talking about what you can do with the data, it's important to understand that QIAGEN has multiple different solutions on the analysis and interpretation space, but specifically looking at what the QIAGEN CLC Genomics Workbench and QIAGEN Ingenuity Pathway Analysis can provide to you. All of these things allow you to look at different areas of the data. At QIAGEN CLC Genomics Workbench, you can do different types of reports, from basic reports to more advanced reports, as well as phylogenetic trees that allow you to look at the virus as it has been identified in different specific clusters. But as discussed, the pathway analysis software in IPA is built on a comprehensive, manually curated knowledge base. Now, this is used by thousands of researchers worldwide and has been cited extensively in studies for the host response to viruses. And here you'll be able to identify COVID pathogenesis pathways targeted for host immune response. Now, QIAGEN has also, for the community, gone forward and actually opened up and provided free CLC and IPA licenses until June 15th to all labs involved in the COVID-19 response. And to get access to this, all you have to do is simply go to this URL and click on this and sign up, and you'll be provided access to whichever tools that you are looking for. But if we look at specifically the QIAGEN CLC Genomics Workbench, it's important to look at what type of workflow that you have and what it actually delivers. Now here we're showing that there's essentially 3 different outputs. One is a basic response, one is an advanced one, and one is a phylogenetic tree. Here we're showing the advanced workflow that delivers coverage, reads, and variants. And you can get not only a summary report, but you can get your coverage maps, output consensus sequences, and then also do variant calling and look at multiple samples at the same time. Now, let's move beyond just talking about the virus and actually explore what else to do when you're looking at a non-biased approach. So QIAGEN has a number of different solutions, as noted, really using its RNA sequencing technologies, and some that have been published on into the public space and protocols.io, looking at RNA sequencing to tackle the virus. If you're looking at greater than 100 nanograms or less than 100 nanograms of input, you essentially have a slightly different approach in how you take that. Less than 100 nanograms, you'll look at the QIAseq FastSelect single-cell RNA kit that preps everything from in the sample. At greater than that amount, you'd use the QIAseq stranded total RNA library kit. Either one of these kits that you use is imperative to knock down ribosomal RNA. As you may know, ribosomal RNA is the most widely expressed RNA that's typically found in a cell. And so it's important to knock that down. But typical approaches in the past have actually looking at just the global ribosomal RNA and knocking that down. The challenge is that as you look across species, especially things that have a combination of mammalian DNA or RNA and bacterial RNA, is that you need to take different approaches to do that. And QIAGEN actually has solutions that target specific ribosomal RNA, whether you're looking at a mammal, whether you're looking at bacteria, whether you're looking at plants or other different types of things. And so here what we're showing is an example where you would actually use both the mammalian ribosomal RNA and globin depletion in combination with our bacterial ribosomal RNA. And the combination of this will allow you to knock down the host and the bacterial ribosomal RNA, especially if you have samples or swabs or areas of the body where you know that there is a considerable microbiome content. Now, as I look at this and provide you an example here, what I'm showing is data of looking at sequencing of no depletion and with depletion of both human reference RNA and as well as gut RNA, and comparing those two results. And so if you look at the bottom left, what you have here is a circle here that shows the amount of ribosomal RNA, gene expression, and unmapped RNA, and the percentage of each as you look upon these different conditions. On the far left, what you see is universal human reference RNA with no depletion. What you see is ribosomal RNA accounts for 91% of the reads. Only 8% of the reads are actually genes of interest. By now comparing that and adding FastSelect, 5S, 16S, 23S, plus the human, mouse, and rat, you're able to now knock down tremendous amount of our ribosomal RNA. So you go from 91% to only 6% of your reads are actually ribosomal RNA. And now this flips this, as the amount of genes of interest are actually represented by 93% of the reads. But as we think about this from a more challenging sample than a reference sample, we have a gut sample that we have, and again, comparing no depletion to using FastSelect. Now, gut is obviously a more diverse sample than a reference sample, but what you see here is something similar. The ribosomal RNA, it accounts for 93% of the reads are ribosomal RNA. Only 6% are actually the target genes that we're probably looking at. But as you add FastSelect here, of both the 5S, 16S, 23S, and the human, mouse, and rat, what you see is completely flipped from 6% of your genes that you're looking at to 82%. And this compares to 8% of the reads are now only ribosomal RNA. So for a more complex, challenging sample, you still see a tremendous amount of uptake and increase of the actual RNA reads. But often the question is, how does this compare in terms of existing approaches? And so, again, with using the universal human reference RNA and comparing that to a few different approaches. One is no treatment, as you saw on the previous slide. The second is using poly-A enrichment, which will enrich specifically for mRNA, which have poly-A tails. And then comparing QIAseq FastSelect to Ribo-Zero Gold, poly-A enrichment, what you see is that enriching for poly-A will still have ribosomal RNA that's found in the sample. And so this background is still not interesting for most experiments. But you typically get between 1.56 and 1.75 or 1.74% of the reads are actually ribosomal RNA from doing poly enrichment. Now, if I compare that to what I'm seeing with QIAseq FastSelect, where I'm not just looking at mRNA, but I'm finding all different types of RNAs that are there. mRNA, long non-coding RNAs with poly-A tails, long non-coding RNAs without poly-A tails, and other RNA of interest, what I'm seeing is a range from 1.46% to 1.68%. So effectively what I've shown is that poly-A enrichment and using FastSelect really demonstrate the same amount of ribosomal RNA that are left over, but now I am seeing the totality of the whole transcriptome, not just with poly-A enrichment. But now as I compare this to Ribo-Zero Gold, and you look at the FastSelect data, again, you're talking about the range between 1.68% and 1.46% of ribosomal RNA that's left with FastSelect. But looking at Ribo-Zero Gold, you have contamination of up to 5.34%, or more than 2.5 times the amount of contamination that you have with using FastSelect. So what you hear is essentially losing reads that you don't have to lose by using FastSelect. By using FastSelect, you can essentially eliminate background noise at a higher degree than you can with any other competitor, or even comparing it to poly enrichment, while still read the entire transcriptome. But as talked about earlier, there are multiple different approaches to looking at host response. And from a targeted approach, there are a few different panel-based approaches that you can choose to take from this. A pathogen that affects a host will have a specific response. And so here you're able to study different key genes, multiple different biological pathways here, in things like toxicology. But if I look at inflammation and immunity transcriptome, I have a whole host of different targets that I'm going to be able to see. Now, the final thing to talk about here is also the immune repertoire, and specifically being able to look at T cells and how they respond to this type of infection. So our QIAseq Immune Repertoire Kit allows you to cover the entire VDJ region of the alpha, beta, delta, and gamma genes. So no matter whether you're looking at the pathways or the T cells themselves, you can hone in on specifically what's going on with these targeted panels. So finally, QIAGEN is delivering assay and analysis solutions to support SARS-CoV-2 research. Whether you're looking to do unbiased testing, you can use QIAseq FastSelect and our host of RNA sequencing solutions. If you're looking to target specific things like the whole viral genome, you can look at the QIAseq SARS-CoV-2 primer panel. You can also look at targeting the host pathways and using next-gen sequencing and also PCR to be able to test these pathways. And finally, all of this data provides you with the ability to now use QIAGEN digital insight solutions, such as QIAGEN CLC Genomics Workbench or QIAGEN Ingenuity Pathway Analysis. So I want to thank you for your time and thank you for listening.